All right, in this video, we'll be looking at tonicity and osmoregulation, divided into two parts because it's pretty big. And so this is part 1 of 2.8. And both of these have to do with the concentration of water and the movement of water. The diffusion of water across a selectively permeable membrane is called osmosis. Osmosis is a passive process typically, and so it is a type of diffusion. Water, remember, moves typically through big protein channels called aquaporins. And so you can see here, there's typically when we think of this, we'll concentrate on the purple dots, right, as having a high concentration of purple and low, and we would expect the purple dots to move, right? But in this case, there's a membrane that prevents the movement of purple dots. Well, there's also a concentration of water here. There is more water on the left than there is on the right, and so we would expect water then to move to where there's a low concentration, thus creating a system of equilibrium. All right, and so this kind of concept is called osmolarity. This has to do with the total solute concentration in a solution. Just a couple reminders. Water has a high solvency ability, meaning that it is a good solvent, typically dissolves things. So solvent is the thing that is dissolving the solute, and the solute is the whatever it is in the solution. So solution is two parts, solvent, solute, and a solution also denotes that there's an even distribution of these things. You typically think of like salt water or sugar water or something like that, in which the solute is dissolved. There's not things floating around in there. And so that's when we use the word solution, we are talking about an even distribution. That brings us to this next point of tonicity. And tonicity is the measurement of relative concentrations between two solutions. This is important because when we use these words, hypo, iso, and a hypertonic, these are comparative terms. We don't say them by themselves. Anytime you use them, you're comparing two things. And that's important. And so usually in our systems, we're going to be looking at the inside of a cell versus the outside of a cell. You can see the picture here, set up some balloons in solution, and the balloon is mimicking the cell. And that's how we're going to look at this. So the internal solutions can be hypo, hyper, or isotonic to the external solutions. Again, we're making a comparison here. And so this is a balloon in an isotonic solution. What that means is that the concentration of solute outside is equal to the concentration of solute inside. And so there's no movement of water, because if the concentration of solute is equal, the concentration of water is also equal. And so there's no movement. In this next picture, you have a balloon in a hypertonic solution. So if the solution is hypertonic, then the inside has less solute, the outside has more solute. And so that word hyper means more. If the solution is hypertonic, it has more solute. That means it has less water. That means that there is more water inside the cell. And so the water inside the cell will move out of the cell, thus shrinking the balloon. A hypotonic solution is the exact opposite. Again, waters will move to equilibrium, and so this causes the balloon to expand. Just a good rule of thumb about this, water will always move from hypotonic to hypertonic. And so water's moving from less solute to more solute every time to get things at equilibrium. Water moves from high to low just like anything else. And so where there's less water, it will move to that solution or move into that other system. Water concentrations and solute concentrations are inversely related. Think about it that way. If there's more solute, there's less water. Water goes from hypo to hyper every time. And water will move independently of solute too. That's important to understand as well. When a cell's in an isotonic situation, what we say is that there's a dynamic equilibrium. And what we mean by dynamic equilibrium is that it's an equilibrium. There's still but it's dynamic in that there's still movement. All right. There's as much water going in as there is going out. And so there's no net movement, but there is still movement of water. It's not like things are frozen. There's still movement. There's just no net movement. All right, so let's talk about how this looks in plant cells. Man, try to get me out of the way. All right. So plant cells, remember, have a cell wall, and so, but they still are trying to maintain this osmolarity. And so, if the environment is hypertonic, that means all the water inside the plant will want to move out of the plant cell. This will cause something called plasmolysis, where plasmolysis is, or plasmolysis is where the cell membrane will actually pull away from the cell wall, and this is not a good situation for a plant. In an isotonic solution, a plant is typically what we'll call flaccid, where the plant is not necessarily pressing out against the cell wall, and it will kind of have a wilted appearance. But what plants want more of is this system called turgid, where the water is there's the outside is hypotonic to the inside, so water is moving into the cell, thus creating that turgid pressure pushing out against the cell walls, which will lift the leaves and cause the plant to be more healthy. This is an important concept with plant because plants are always trying to maintain this pressure so that they can, do photosynthesis, do their normal processes, and very, very important sort of thing. This is the optimal, again, this turgid state is the optimal state for a plant. And in animal cells, osmoregulation maintains water balance by allowing the internal solutions, by determining that internal solution versus the outside solution as well. But animal cells have a problem in that they don't have cell walls. And so if the external environment is hypertonic, then that will cause water to move out of the cell, and that will cause the cell to shrivel. That's not very good. If the outside is hypotonic, then that can cause a cell to lyse. And so in a normal situation, there is isotonic, which means that the cellular concentration is the same as external cellular concentration, which means that there's no net movement. And this would be more of the ideal situation for animal cells.